Hmm. Okay, so we'll get started on this. Okay, welcome to week 12 of NPTEL Computational Systems Biology. So this is the last module of the course of week 12. So yeah, and tomorrow I'll be taking an extra session of 2 hours to recap what has been learned in the course and we practice some problems as well. So yeah, today's class is going to be considerably shorter because the content is much less compared to other weeks. But yeah, nevertheless, we'll go through it. Okay. 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 So yeah. So we'll start by talking about neural networks. Sorry, neutral networks or non-neural networks. Those are different things. Okay. So what actually is a neutral network? So you consider yourself a discrete macromolecular sequence of genotypes of n monomers, such as RNA or protein. You consider that as a space where everything is distributed, and in this space, two sequences are immediate neighbors if they can be transformed into one another by a single point mutation that changes only one monomer. So you have this set of monomers in the space, and how are they related to each other is basically one is a mutant of the other. So if there's one slight change in the DNA or the RNA of the protein or the amino acid sequence, one can be converted to the other. So these are what are known as immediate neighbors. So they are transformed into one another by single point mutation, and it, it can change and changes only one monomer. A genotype network, which is called the neutral network, is a connected set of genotypes that produce the same phenotype. So you have this connected set of, like, so for example, you have a whole gene and that is producing a particular phenotype. Now that gene has been mutated a slight bit, but the idea is if you mutate it in, say, probably intronic or intergenic regions of the gene, so it will not change the protein formation, the amino acid that the gene is going to be, and the amino acids, the RNA is going to be forming from the gene, and ultimately the phenotype the individual or the organism will exhibit. Because the difference between the wild type gene and the mutant gene is very minor and not very functional. So these can be clubbed together. So these actually are set of would be connected genotypes that can produce the same phenotype. So that is what a genotype network or a neutral network means. So within a genotype network, these genotypes are directly connected to each other if they differ by small mutational changes, as I was saying. So if you have small changes, small mutations, and non-significant in terms of their functional consequences, uh, they can be directly connected to each other. Uh, so if it's only a single base pair change or a single base pair deletion or insertion or something like that, they can be connected together. Now, these genotype networks are thought to be common organizational property of genotype spaces of biological systems at different levels. So, uh, and there has been ample empirical evidence for their existence to support the notion for RNAs, proteins, uh, or binding sites of regulatory proteins, etc., to be grouped under the similar concept of a genotype network. Now, uh, the exploration of such networks became possible only with the ability to characterize molecular phenotypes from many different genotypes, either computationally or experimentally, which is the concept of GWAS, or concept of doing sequencing and understanding the mutations that are actually contributing to the phenotype. So you need experiments like this, or you need data like this to create these neutral maps, or these neutral networks of genotypes, which are actually distinct genotypes, but they can form a similar type of phenotype. Uh, yeah. So this was the initial work on of the neutral networks, or disconnected genotypes. Genotype networks was done mainly using RNA secondary structures. So basically, they showed that the people working on it showed that the neutral networks associated with many individual RNA secondary structures are vast and span the sequence space. And the concept was later applied to proteins and to higher order molecular systems such as regulatory networks as well. So mm, now the idea of robustness is also derived from this concept of neutral networks. So think about it. You have different types of genotypes that are producing the same or similar kind of a phenotype. Now, the idea of robustness is that if the system is in a steady state, if it is in a stable steady state, if you push the system slightly to any side, it will come back to its original steady state. That is what a robustness means. Robustness or resilience, can use them interchangeably, but that is what it generally means. Now, uh, the idea is that you have these small mutations. If you imagine you have small mutations in the gene, but the ultimate product is not getting hampered. So that means the the system is very robust. So even if you mutate certain positions of the gene, the phenotype exhibited will be the same. So that idea is also drawn from the concept of these neutral maps, that you have this set of genotypes that are clustered together or that are connected together as a large connected component of the network, which are actually differing by each other from each other by single point mutations or small insertion deletions, and they are producing the same phenotype. That is the whole idea of robustness, right? So you have these mutations, but still you are producing the same phenotype. So yeah, and so these are two set of panels of the diagrams, which the first one is showing low robustness, and the second one is showing high robustness. In each of these, in each of the network, the rectangle represents a part of space of genotypes. The gray circles correspond to individual genotypes on a neutral network, and the straight line links to genotypes. They can be converted uh, through a single point mutation. Sorry. The top three panels actually show neutral evolution, show sorry, how neutral evolution explores genotype space for a phenotype with low robustness. The phenotype with a small neutral network in which the individual genotypes have, on an average, few neutral neighbors. So, so basically, neutral evolution is basically how does this evolution occur? So you have uh, you start off with a set of genes so basically you have your genome. So over time will have mutations in the genome. So the mutations which are going to help adapt uh, to the environment of that particular species, or be it human or other species, that mutations will propagate and be selected for the further generations, and the rest of the mutations will be discarded. So the similar concept is there. So if you have if you're just moving, if you start from the middle and you start mutating bit by small mutational changes of the nodes surrounding the center, so you'll move away slightly in terms of the genotype, but the phenotypes is going to remain the same. So if you move away slightly and the phenotype changes, so it is a sudden change and it cannot be sustained by evolution. So what evolution will do is baby steps. So you have a slight mutation, the phenotype is actually almost the same, maybe slight difference, or even if you do not see a difference, that is also fine. So you have this multiple generations of these mutations accumulated to actually show a shift in the phenotypic distribution from what we had before. Okay, so yeah, so this is a low robustness condition where basically after you move from one node to another, the phenotypes are getting changed. And the differences, the bottom three panels show the same evolution process, but for a robust phenotype. Uh, all panels contain the same number of genotypes to facilitate comparison. They therefore do not reflect, sorry, yeah, so they therefore do not reflect neutral network sizes, but merely the fact that genotypes on large neutral networks also tend to have more neutral neighbors. These blue circles correspond to individual members in the population, and these red stubs are actually hypothetical deleterious mutations that cause a mutation to the genotype lying outside the neutral network. The differences in the number of red stubs between the top and the bottom panels actually is an indication of how robust actually the individual networks are. So the idea is that the robust phenotypes with large neutral networks are subject to fewer deleterious mutations, and the vice versa is true as well. So if you have large neutral networks that are subject to more deleterious mutations, so it is going to be a less robust phenotype. And yeah, so the two leftmost panels show two genetically identical populations. with moderate genotype diversity, in which several individuals have the same genotype. Uh, as these populations evolve through mutation and selection, confining them to a network, the population with the robust phenotype will spread more rapidly because fewer deleterious mutations will impede the accumulation of genotypic diversity. Right? So that is a general idea about what are neutral networks and these genotype-phenotype maps that we are going to talk about in the next slides. Okay, so what is the relationship between uh, this neutralism or robustness in terms of phenotype and selectionism in terms of evolution? Now, selectionism is supported by patterns of genomic evolution, and the importance of robustness and neutral change is uh, can be highlighted by molecular phenotypes. Now, how to reconcile these two observations in the form of a conceptual model that can be centered on the neutral network metaphor, right? Say, for example, first you take a single genotype that undergoes a random mutational change. A random walk, so we talked about random walks and Eulerian networks. I hope you remember that. So a random walk on the neutral network of the phenotype, it adapts, right? So you start with a genotype. It'll undergo a simple small mutational change, which is basically a random walk on the neutral network of the phenotype it adopts. Assume that this phenotype is suboptimal and the target of the evolutionary search is a better optimal phenotype. Now, what will happen? The sequence might first take several neutral mutation steps on the neutral network, focus on neutral change. So, that is very important. The changes are mostly neutral, although these deleterious changes may also happen, which can be likely, but then it'll act against the selection mechanism, right? So it will be discarded if too many of these deleterious variants actually come out. Right? So after some steps, sorry, so yeah, after some steps, a phenotype changing beneficial mutation might produce a new phenotype closer to the target phenotype. A random walker will thus hop from the first to a second neutral network. From then on, the cycle repeats. A number of neutral mutations and exploration of a current neutral network would be followed by a mutation that discovers a new phenotype closer to the target. Right? Just give me a second. So, yeah, we were talking about the relationship between the neutralism and selectionism. That you have this small baby steps of neutralism that can ultimately lead to selectionism. So you can move in the direction of selection. You can move in the direction of the selection by the small random mutations, which will produce the same phenotype as your unmutated gene or unmutated genome, and taking these small baby steps of uh, neutral changes can lead to discovery of a newer phenotype, which is closer to the target of your deleterious mutations. Okay, so yeah, this is a very good example of this. So these are actually different nodes are denoting different genotypes, and the connections between these nodes are basically if one point mutation can convert uh, uh, one genotype to the other, right? So yeah, so these all these circles are genotypes. So as yeah, the path of only a single genotype through sequence space is shown. So this genotype starts from here and moves to here, then here, and then ultimately here. Right. The genotype evolves through a hypothetical adaptive phenotype, which is not shown here. In order to arrive at this phenotype, it traverses through four different neutral networks. These are actually individual neutral networks of its own. Now, from going from one phenotype to a slight change in the phenotype, it had to pass through four of these different neutral networks. Uh, uh, yeah, Within each of these neutral networks, evolution is neutral, but at the transition between the neutral networks, positive selection occurs. So that is the idea. So you have this imagine the concept of modularity that we discussed earlier. So you have the whole genotype network, all the possible combinations of the genotype. Now you have modules inside this network. So each of these modules represent phenotypes which are very close to each other, and one point mutation can convert one genotype to the other. Now inside these, if you have a set of nodes, so if you are moving from one node to another, the genotype changes slightly, but the phenotype remains the same. So basically, that's a neutral mm, transition, basically, right? But if you say, imagine these modules that we are talking about, if you move from one module to another module, that is when positive selection occurs. So that trait is actually selected and passed on to the next generation. So this is actually quite intuitive in terms of understanding evolution and predicting neutral and beneficial changes, right? So mostly, if positive selection is occurring, mostly the changes are going to be beneficial as well. Excuse me. Yeah, okay. So we have three types of. So now we'll talk about this genotype phenotype map. So now we talked about neutral maps. Those are basically changing genotype without changing phenotype. Now we are seeing how changing genotype will also change phenotype when you move from one transition from one module to the other inside the network. But now we'll talk about genotype phenotype maps. So what is it? It basically describes the relationship between the genotype space and the phenotype space. So your genotype space is two-dimensional and your phenotype space is a single dimension. So together it makes a three-dimensional kind of a plot. Yeah, the entire surface represents a hypothetical global genotype phenotype map relating all possible genotypes to the phenotype. Right? Ellipses illustrate local genotype phenotype maps in which deduce bias based on variation in population, and the genotype phenotype map concept can also be extended to the description of physiological, developmental, and functional mechanisms by which these genotypes direct the epigenesis of phenotypes, which is the mechanistic genotype phenotype. Right? And the populations evolve by moving. So what happens is they move into the genotype space, which change the phenotype and the structure of the local genotype phenotype map, and that is how evolution occurs. And yeah, so if you see in this figure, the global map shows a discontinuity in phenotype space, whereas yeah, such as the genotypes on the other side of the boundary are labeled as structural change. Right? And they can show discrete differences between the individual phenotypes. And these differences are represented by the two panels on the right. So these are the figures, and the process of epigenesis proceeds like a ball rolling a downhill on each landscape. So this is what is the concept of Waddington's landscape. You have the genotype phenotype maps, and there is a there's kind of this surface on which the ball of probability actually rolls on, and whenever it goes, wherever it ends up, the phenotype you will you will get wherever it ends up. The maps in the front of the structural change boundary will reliably produce one of the four phenotypes at the bottom of each channel. Uh, okay. The idea is that if you define GP maps, genotype phenotype maps like this, the global genotype phenotype map of the overall individual or the species is mostly going to be constant, and it technically will not evolve. The the local genotype phenotype map evolves along with the genetic changes in the population, and the mechanistic genotype phenotype map evolves when the populations cross the structural change limit. Okay, so this is an example that we'll be talking about here of the genotype phenotype map in case of sulfur metabolism. So you have these metabolic reactions, metabolic networks on the left hand side. No, okay. So there are four major networks here. So the first network is the metabolic network, which actually will tell you which metabolites are interacting and reacting with each other to form what. And then the genotype network, which is some gene mutated or anything is happening there, that is there. And the genotypes is the list of categorized or catalyzed reactions, and the phenotype is the probability does it actually survive on one carbon source. So the genotype phenotype map of metabolic networks is this is how it looks. So different representations of a hypothetical metabolic network will look like this. So first will be as a node in the genotype network, or as a binary vector, or as a listings the net reactions in the network. Each genotype on the genotype network has around 1221 neighbors. So this is from the real data that has been studied. So yeah, each genotype will have 1200 neighbors. Now, these 1200 neighbors are part of the neutral map, right? So these will have genotypes which are slightly different from the original one, but yeah, they will be differing by single point mutations, but the phenotype produced will be very similar. Neighbors in the genotype network are connected by edges, and the colors of the genotypes will are represented by different phenotypes. The phenotypes of the metabolic network are computed using flux balance analysis applied to 124 environments with different sulfur sources. Two hypothetical phenotypes are represented as binary vectors. Listing the environments as a genotype is viable in there. The random evolutionary walks can be seen as a path on a genotype network that stay on genotypes with the same phenotype, and mutations correspond to additions or deletions of individual reactions from the metabolic network. And yeah, and the total number of genotypes is around 21,000 here. So here is the uh, whole learning from this robustness, neutrality, neutral maps, and so on. Okay. So you have high dimensionality adaptive landscapes. Just give me a second. Okay, so ultimately it comes down to this. So you have high dimensionality adaptive landscapes, which are characterized by large neutral areas. So if you consider the whole this as a workflow like this, so high dimensionality adaptive landscapes, they are characterized by large neutral areas. So large neutral areas meaning large number of nodes that are represented by genotypes, which are which are actually point mutations compared to each other, and they produce a similar phenotype. Now, these are critical to evolvability, as we were talking about. These are critical to evolvability. The efficiency of an evolving system at finding beneficial mutations or recombinants, right? And now this is facilitated by genetic networks of hub and spoke type. So hub and spoke, basically you have a hub in the middle, which is basically the core group of nodes that are actually maintaining the system, and you have a lot of edges connecting to those nodes, so that which means that those nodes are the most important parts there. Uh, yeah. Now, if you move in from here, that is one concept. So you have high dimensionality adaptive landscape characterized by neutral areas, which is critical to evolvability and so on. Right, now we start from here. Modularity, which is the degree of interconnectedness of a system. That is also important because it mediates between the evolvability and robustness. So what is robustness again? The property that allows a system to maintain functionality despite internal or external perturbations. So the internal perturbations will be genetic mutations, and the external perturbations will be environmental factors. Right? So, mm, yeah. So, and these will be arising from uh, feedback regulatory systems, which is again based on the hub and spoke type genetic networks. Now, natural selection, on the other hand, is also very important. Now, if natural selection can change your modularity, natural selection can indirectly affect your evolvability and robustness. And yeah, and so natural selection is kind of a mother load that actually is controlling a lot of things directly or indirectly. So a lot of things actually depend on selection procedure because depending on the mutations. Uh, if you go by this, so you have this set of modules. So the yellow part is one module, the green part is sorry, blue part is one module, the red part is one module, and the green part is another module. So inside each of these modules, the mm, so in sorry, I just to join. Yeah. So inside each of these modules, the transition from one node, the movement from one node to another is basically your moving point mutations. Inside that module, the evolution is neutral. But if you move from this module to this module, so this to this to this here. So if you move from one module to the another module, there the positive selection needs to occur. So selection actually can hamper the evolvability and the robustness of the system. And now these two are also inversely related, but for mediation of modularity. Right. So this is actually the whole system, how the system looks like in terms of modularity, how it is contributing to evolvability and robustness, and this evolvability, how it is actually a kind of a function of high dimensionality adaptive landscapes. And natural selection is kind of presiding over everyone uh, in terms of regulation. So if you characterize this as a network, this is also quite a complicated network in terms of each of the nodes making representing a functionality. Right. So this is also this kind of is the overview of the whole thing. The major things that we have talked about in this course, basically modularity, uh, um, robustness, resilience, etc., etc. So this slide kind of sums it up. So yeah, uh, in terms of uh, in terms of content, that is it. But we have a few questions to solve, so I'll put this on the screen for 2 minutes and then we can discuss the answers. Yeah, you can write it and write the answers in the comments if you are solving it. So, okay, let's discuss. Okay, so you have these four networks. So in the first, so the question is, you have to arrange this in terms of increasing network robustness. Uh, Network robustness is basically the network's ability to maintain its overall structure and function despite the perturbations. Uh, um, like the idea is if you have more connections and alternative paths between nodes, they actually indicate higher robustness. So if you consider the first network, this is actually a relatively sparse structure and it has very few connections between the nodes compared to the other networks. The second one is also less connected, but it is still more connected than the first one. The third one is actually more connected than the first and the second as well. It has a high density of connections, and also there is some sort of interconnectedness between them as well. And if you consider this the red node as a critical genotype, but the overall network will still have alternative paths. The fourth one, it has the highest density of connections from visually understanding. Which means there are many alternative routes between different parts of the network, which is actually what we are looking for. In terms of increasing robustness from the least to the most robust, the least will be the first one, then two, then three, then four. So the answer is one is the least, two, this is a little bit more, third is a little bit even more, and fourth is the highest. Now, the logic behind it is that like the more sparse you are, the less connections that you have, the less robust you are going to be. Because if you shut out one node or delete one node, it is very unlikely that uh, uh, you can reach the so you can travel from one node to another. From say, for example, you remove this particular node, right? You can still travel from this node to this node, right? But if you say, for example, remove this node in between, you cannot travel from this node to this node. So basically, the idea is the more connected you are, the more alternative paths you have, and the more robust your network will be. Okay, moving on. Okay, uh, so let's discuss this. Okay, so the question is arrange the networks in order of increasing evolvability of the network. So evolvability basically is basically kind of a balance between robustness and flexibility. You want enough interconnectedness to like kind of buffer detrimental mutations, which is the robustness part, but also enough degrees of freedom or potential for new variations to arise to be explored, which might be hindered by excessive interconnectedness. So you need an ideal kind of a scenario there. So in this case, this mutation, the fourth uh, network, if you notice, you have a disconnected node here, and also it is very highly connected. So the idea, sorry, the idea is that the most robust, and also there is a option for new variations as well. So fourth is having the highest, fourth is having the lowest evolvability because it's most connected and it only has this one single point where it can evolve, right? Or as two, right? These two marked in light green, right? Next, you have this first, the second, and the third. So next is in terms of evolvability, less connections, it's more, but it does not have enough robustness to actually move through. uh, mm, the thing is, it has a lot of potential in terms of getting a new mutation, but it does not have enough robustness to kind of support that new mutation, kind of like it will be a drastic change, deleterious change, and it can lead to not positive selection, right? So the same concept is applied to 2 and 3. So 2 and 3 will be slightly better than 1, and 3 being the best. So so 4 is the lowest one, then it's 1, then it's 2, then it's 3. 3 is the best network to evolve. So you have enough amount of robustness inside, and you have these 3 connected nodes which are actually going to allow you uh, for evolvability. Why 4 is not counted as in the same picture as 3? Because you have this disconnected node here. So that is an issue. So evolvability is going to be severely hampered there. And in the rest of them, you see all have 3 of these nodes. See, except for the fourth option, and one of the node is also not connected. Right? Okay, moving on. Okay, uh, let's discuss this. Okay, so again, what is a neutral network? It is basically a landscape of genotypes where many genotypes can produce the same phenotype. That is what a neutral network is. Now, these genotypes are connected in the network if they are just one mutation away from each other, as we discussed. And now, the first statement is the neighbor nodes in the neutral network exhibit different phenotypes. This is incorrect because, by definition, genotypes within a neutral network should exhibit the same phenotype. So the first one is incorrect. The second one, a neutral network is a set of genotypes that share the same phenotype. This is correct. This is actually the definition of neutral network. The third one is a neutral network is a set of phenotypes that share the same genotype. This is actually incorrect. It is actually the other way around. It is the a set of genotypes that actually share a phenotype. And the final option is the neutral nodes in the neutral network exhibit different genotype. This is correct. Neighboring nodes in the network represent the genotypes that are 1 point mutation apart. So they must have slightly different genetic sequences as well. So that is correct. So the answers are the second and the fourth option. Right? Oh, okay. Sorry, I missed your doubts. So first of all, more connected are more robust. Yeah, that is true. If the more connection is there, so imagine you have a fixed set of nodes, but if you have multiple connections between two nodes, if one of the connection is somehow cut off, you have other connections to rely upon, right? Uh, yeah, so the answer was not the same for question two I discussed. No, but the third question you're correct. Options two and four are correct. That is right. Okay, um, let's discuss. So it's not that difficult. So you have, if you can see it like this. Wait, I think it is opposite. So basically, I'll tell you. So length of each of the peptide, it's given as 25. Length of the peptide, right? 25. Now you are told, where's the question? Length of the peptide is 25. Now you are told uh, to consider to get the size of the genotype space in which the protein will be evolved by a single amino acid change. Now you have 25, the length of your peptide is 25, and at each of those 25 positions you could have 20 amino acids because there are 20 essential amino acids. So the total number of possible combinations will be 20 to the power 25. Now you just need to kind of simplify it, right? Because you have to give the answer in like this format, right? So you need to simplify 20 to the power 25. So you can break it down like this: 2 to the power 25 and 10 to the power 25. You can calculate the 2 to the power 25 separately. It comes as around, if you like simplify this, so it becomes 2 to the power 10 whole square into 2 to the power 5. So it becomes 32 into 10 to the power 6. If you replace this here, so 32 into 10 to the power 6 into 10 to the power 25. So it becomes 32 into 10 to the power 41. Sorry, 31. So 3.2 into 10 to the power 32. So this is the answer. The answer is not very like informative. The main idea is that you have this 25 positions of amino acids, and each position you could have 20 amino acids. So it's 20 to the power 25. After that, you just need to do a little bit of simplification. That's all. I hope this is clear as well. There's no formula. There's no formula. So just imagine you have 25 positions. In each position, you can have 20 amino acids, right? Because there are total essential 20 amino acids that make up all the proteins in our body. So the total number of combinations, it will be 20 to the power 25. Then you just need to do the simplification. 20 to the power 25, you can write 2 into 10 in bracket to the power 25. Now you can separate out 2 to the power 25 into 10 to the power 25. Then you simplify 2 to the power 25, it comes as around 32 into 10 to the power 6. And if you simplify that, it becomes 3.2 into 10 to the power 31. 32. It's just like a kind of a simplification process. The main idea is that it's 20 to the power 25. You have 20 amino acids that can be possible in one position, and you have 25 of this sequence. So 20 to the power 25. Formula, you can, there's no formula by the way. You can see this if you can understand. I think it is coming as reverse, but if not, uh, you can understand. If you can look here, you can understand. So you have to replace. You have to put this here in place of 2 to the power 25. That's all. Nothing else. I hope it is clear. Okay, let's move on. If you have any doubts, please ask. Yeah, that's all. Just that, then you simplify. It's just that they've asked the answer in this format. That is why we are simplifying. They've asked the answer in this format. That's all. Nothing else. It's just 20 to the power 25. Okay, what does actually degree of any genotype in the genotype space mean? It refers to basically the number of other genotypes that are only one amino acid change away from a given genotype. So there are 25 positions in the amino acid in the peptide, and for each positions there are 19 possible amino acid changes, right? Because one will be there and can be replaced by 19 other. So the degree of any genotype would be 25 into 19, 25 positions, 19 changes. So that is 475. The answer is 475. I hope this is clear. This is all like pretty simple. So you have 20 positions, you can have 19 replacements for every position. That will be your uh, total degree, right?
So there are 25 amino acids, right? So from where the 20 number came from?
20, oh, sorry. Yeah. So you have 20 amino acids, right? In your body, every protein is made up of amino acid. How many total amino acids are there in your body? 20 essential amino acids. So if there is one amino acid and that is getting replaced, it can be replaced by 19 other. So in 25 positions, you can have in each position, you can replace it by 19 other amino acids. So it's basically 25 into 19 for the total number of uh, uh, replacements. So that is where from 19 is coming. Is it clear now?
Yes, so it's basically the number of possible amino acids.
Yeah. It's basically the number of possible edges they're asking. So you have one genotype. Around the genotype, you'll so there's one node. Inside the node, there is one genotype, and there are other nodes surrounding that particular node, which are just one amino acid away from this particular genotype. So every node has one amino acid change from this genotype. Every node. Now, what the question is asking is, what is the total number of degrees of these nodes? Base total number of edges connecting these nodes. So you have 25 positions in the peptide, and each position will be surrounded by 19 such nodes. So it's 25 into 19. I hope that is clear.
Degree of connectivity.
Sorry?
So it's basically like a degree, degree of connectivity.
Yeah, yeah. That is what it is asking. Degree of connectivity. Yeah. Is it okay now?
Yes.
Oh.
So for the previous answer, I solved and it coming 3.3 E2. Can you show that solution again?
Uh, okay. I think I have the solution written. Just give me a moment. I'll paste it here. Okay, I think you can see it from here. Only. One second. Can you see this? So the question was the question number 3, right? So sorry, question number
Four.
4. 4, right?
Four.
This one, right? So you are asked like what is the genotype space for this? If you have a peptide of length 25, and you are assuming the edges to be the protein evolved by the change in the single amino acid. Okay, so we are dealing with a peptide of length 25. Each position in this peptide can be occupied by one of the 20 standard amino acids. So for the first position, there are 20 possibilities. For the second, there are 20, and so on for 25 positions. So the total number of possible peptide sequences, which in this case are the genotypes, we need to multiply the total number of possibilities for each position together. That is how you get the 20 to the power 25. So this is should be 20 to the power 25 like this. If you calculate this, so these are all like to the power. So if you can break down 20 to the power 25 in you can break down this 20 as 2 into 10, right? To the power 25. right? And then I think something is wrong here. So 20 to the power 25, 2 into 10 to the power 20. This is fine. Yeah. So you're just breaking down the 20. 2 into 10 to the power 25. So this becomes 2 to the power 25 into 10 to the power 25. Till here it is fine?
Yeah.
Yeah. Now you calculate this 2 to the power 10. So if you do 2 to the power 10, it is approximately 1024, which is approximately 10 to the power 3, right? So this you have calculated as around or approximately 10 to the power 3. Now 2 to the power 25. So this part 2 to the power 25 is 2 to the power uh, uh, 1 second. Yeah. So this is 2 to the power 25 equal to 2 to the power 20 into 2 to the power 5. Right. So 2 to the power 25 also you are breaking it down. 2 to the power 20 into 2 to the power 5. Now this can be represented as 2 to the power 10 square. This is 10 square into 32. 2 to the power 5 is 32. So 2 to the power 20 you are writing as 2 to the power 10 whole square into 32. So this becomes uh, uh, 10 to the power 3 ka whole square into 32, which is 32 into 10 to the power 6. Therefore, 20 to the power 25 will be 32 into 10 to the power 6 into 10 to the power 25 is equal to 32 into 10 to the power 31, which is 3.2 into 10 to the power 32. Is it okay? It's just a simplification. There is no uh, uh, caveat in this. Just you are just simplifying. First, you break down this 20 into 2 to the power 10. Then you simplify this as 2 to the power 25 into 10 to the power 25. Then you do the substitution. 2 to the power 25, you calculate what it is, 10 to the power uh, 32 into 10 to the power 6. And you just replace 2 to the power 25 here, right? Just replace 2 to the power 25 here. So it becomes 32 into 10 to the power 31, which is 3.2 into 10 to the power 32.
Got it. I
Okay.
think the round of error, I think 3.4.
This thing, I think you rounded off a little lower, so that is why it is coming. Yeah. Okay. Okay. This we did. Now, yeah. Okay, let's look at the answers. Okay, so the question is, which of the following statements are true about robustness and evolvability? So the first option is the phenotype robustness and phenotype evolvability are typically positively correlated. That is true. The more robust your phenotype is, the more evolvable it is. Because the idea is that you can have these small neutral changes in these if your network is very robust, but it would not drastically change your phenotype. And these small changes actually bring about evolvability. It pushes you to a certain phenotype. If you have a drastic change, positive selection won't work. So typically, phenotype evolvability and robustness are positively correlated. If that is true, the second option is incorrect, which is negatively correlated. So going past that, third option, the average genotype degree of a neutral network is directly proportional to the phenotype robustness, which is true because the more degrees you have, the more robust is your network because you can have multiple paths to travel from one node to another, right? Even if you delete one of the paths, you can still travel if your network is robust and it has a high degree. If it has a high degree, it is robust, right? So that is also true. And generally, robust phenotypes exhibit higher evolvability, which is actually the same as the first option. But yeah, this is also true. So first, third, and fourth options are correct. Uh, yeah. Do you have any question?
So higher the robustness, higher the chances for evolvability, right?
Yeah. So I don't think you had joined it at this position, but this is the idea. So if you have a lot of robustness inside the network, you can move from one place to one. So you can mutate from one node to another node. But in terms of the phenotype change, in terms of neutral map, your phenotype change is very minimal. So if your network is very robust, it will do is it will resist this change in phenotype. So that is what is the key contributor to evolvability. Ultimately, in evolvability, when evolution will occur, your phenotype will change, but that change cannot be drastic. It needs to be a small step by step, which is actually facilitated by the robustness of the network. Is,
So it is same for genotype also.
it is same for genotype also. Actually, I'm talking in context of genotype only.
Okay.
Yeah. Yeah, so that's it from my side today. Yeah, this is the last question. Tomorrow I'll do a course recap. So I've made the presentation for that. I'll discuss actually all the 12 weeks starting like the major topics. I'll discuss it just to give all of you a recap so that when is your exam? I don't know when is your exam.
I think on 4 May 4, I think.
May 4. Okay. So that is
Altogether
for
not released, but expected was May 4.
Okay, okay. So yeah, so tomorrow will be the extra session. Well, I'll kind of recap the whole course and yeah, you can join, but it will be from 5 to 7 instead of 6 to 8 that we generally have here. So keep
Yes,
that
sir,
in
I
mind.
saw that mail. Right.
Sorry?
Yeah, I saw that mail just
Okay,
now.
okay, okay, okay. You can join and we'll be discussing almost the whole course across the 12 weeks. Okay?
Okay. So regarding that,
Yeah.
these PPTs from week 9 onwards, it's not uploaded, and for 7 and 8 also, it is not uploaded.
Are you sure? Because uh, I, I actually let me check.
Yeah. The attendance is uploaded, but
Oh, okay. One, two,
7,
three, four,
7, 8.
5, 6, 7, 8 is missing. 9 is there.
Mm.
10, 11, 12 needs to
Mm.
be uploaded. Okay, okay. I'll upload. I'll upload actually right now. Oh, I
Okay.
missed this. Okay, okay. I'll do that.
Okay. Mm.
Okay. Any questions?
No, no.
Okay, okay. Thank you. Thank you for joining. See you tomorrow.
Thank you, sir, for the session.
Yeah, thank you.
Bye.